Welcome back to class time. It's now time for Cape Biology. I am your teacher, Joanna George Johnson, and today's lesson will be on respiration. All right, let's begin. So we have here respiration. Most of you would be familiar with respiration already because you would have done it at CSEC level. But however, just like with photosynthesis, it would be very simple at the CSEC level. At the CAPE level, we go into a lot more detail. The whole biochemical process of it is what we will be looking at today. So these are our objectives for today. We will also be looking at the structure of the mitochondria as well. So let's get to it. Lots of the time, the respiration has a lot of misconception. So I need to throw some of these things out from now before we even get into it. Because a lot of times we ask students about respiration, they think it only occurs in animal cells. Of course it doesn't. It occurs in all cells, animal or plants, bacteria, any type of living organism must undergo respiration. Sometimes students think it's related to breathing. Well, somewhat. But remember I just said to you that all cells undergo respiration, but not all organisms respire. So that would mean that there are some organisms that need to respire, clearly, and they are not breathing. So it is not totally related to breathing, but in humans it sort of is. We'll explain that later. Respiration really is the release of energy from our food or whatever nutrients we take in, mainly our carbohydrates, all right? And it is a release of energy in the form of ATP. All right, and so therefore, we need to recognize the main impact or the main purpose of respiration is about ATP. Next thing is that it occurs in the mitochondria of cells, and usually I say animal cells. Most children tend to think this. Now, class. First things first, I know you're going to say, oh, but miss, everybody tell me already that the mitochondria is a powerhouse of the cell. And it is. It is. However, first of all, it starts in the cytoplasm. All respiration starts in the cytoplasm. And the main reason for that, if you remember from Unit 1 days, that different cells, there are different types of cells that exist in nature, and some of these cells don't have large cellular organelles, internal cellular organelles, such as a mitochondria. So if all cells respire, and not all cells have mitochondria, so therefore how it is that respiration must occur in the mitochondria? No, it doesn't. It actually starts out in the cytosol. Sometimes you say cytoplasm, but if oxygen is around and the organism has the capacity to, then it will continue in the mitochondria. And yes, there is mitochondria in plant cells as well. Sometimes students tend to mess that one up as well. And also there are times when students think that if it occurs in plant cells, it only occurs at night. No, it occurs all the time. Remember I said to you that the point of respiration is to produce ATP. And the problem is with ATP is that ATP cannot be stored by the body or by the cells, and so therefore there needs to be a constant replenishment of ATP in the cell. And so therefore, if we were to stop respiring right now, you would not have been able to hear the end of this sentence because you would have run out of ATP, and because of that, you would not be alive. The importance of ATP. First, it fuels all metabolic or all chemical reactions that occurs in the body or in the cells. All right? So it's called the energy currency for all living cells. Whether you are a bacterial cell, or a cell in a mango tree or a breadfruit tree, or a cell in a cat, a frog, a dog, a cat, or us, still ATP that is being used as the energy currency. It allows for the movement of all living cells For example, from the wiggly of the sperm cell up the fallopian tube to us doing the newest dance move, all of that requires ATP. And so therefore, you see how important it becomes to all living cells. It also allows for active transport in cells, as you should be aware of. Now, typically, oh dear me. Typically, anyway, let's move on. The respiration in our cells tend to come in several steps. Typically in grade 11 or grade 10, when you would have learned respiration first, what would have happened is that you would have learned an equation, which hopefully I can remember, where it starts out with glucose, okay, plus oxygen going to give you CO2 plus water. And we know ATP is given off. Now, this looks like a very simple equation, but you have to remember something, that if you look closely, those of you who do chemistry especially, you will recognize that this equation is actually a combustion reaction. And those of you who don't know what combustion is, that is a reaction that causes a fire to take place. Clearly, our cells don't have fire going on each time. So this equation doesn't really happen in our cells, because then we would not survive it. So what happens is that the process of respiration happens in increments, so that all of the energy is not released at once, as in a combustion reaction. Instead, what happens is it is released at different stages. And over here we see where we have several stages of what we call aerobic respiration. This is the respiration that occurs in organisms that have a mitochondria and that oxygen is readily available. All right? So we are going to be looking at the different stages, but today we are only going to be focusing on glycolysis, mainly because it is a long process, and it's the process that occurs in all living cells, whether or not you have a mitochondria. The reason for this is because glycolysis takes place in the cytoplasm, and it also takes place in all living cells. Bacterial cells, eukaryotic cells, prokaryotic cells, all living cells will undergo the process of glycolysis. Okay? So let us move on. I'm going to start that whole thing now. Now, the word glycolysis literally means splitting sugar, or sugar splitting. Glyco speaks to sugar, and lysis means to split, so it's called sugar splitting. All right? It is an enzyme controlled process, as you know, as we spoke about earlier, that the process of respiration, aerobic respiration, that is respiration that uses oxygen, actually is about combustion. And because we cannot have fire going off intermittently in our cells, because that will be spell disaster, what happens instead is that enzymes are used to control the process. So this particular process is very important, and so therefore you find lots of enzymes are involved. Luckily, you do not have to learn all of the names of each of the enzymes, and so that's great for you guys, okay? Back in the day, we had to. It occurs in 4 major steps, so we will be discussing each of these 4 major steps, and we are going to talk about the final products of respiration or glycolysis, and we'll talk about the overall yield. So we are basically, this is the summary before we even start. So let's get to it at the first step of glycolysis. So the first step is called the activation phase of the glucose. Now it's also the energy investment stage. A lot of books will tell you it's called the energy investment stage, mainly because even though respiration is about the making of ATP, pretty much, it also involves, at the start of it, the investing of actually 2 ATP molecules to the process. Okay, so you know, you have the old saying, you have to spend money to make money. So this is the money being spent, the ATP molecule being broken down to form ADP twice during the first stage of glycolysis. It entails the addition of a phosphate group to the glucose molecule. Now, what you notice that it's the phosphate group is added to the glucose. Remember, glucose is a 6-carbon compound. It's very important that when you're dealing with organic type of processes, especially when it's not a one-step type of reaction, but it's a process, you need to always remember and focus on how the number of carbons are going. Very important. So therefore, first of all, you have the phosphate group being added to the glucose to make glucose 6-phosphate. And then there's some isomerization that goes on to convert the glucose into fructose 6-phosphate. And then a next addition of the ATP, again, calls fructose 1,6-diphosphate. So there are 2 phosphate groups attached at either end of the glucose molecule. Please note that some books will say fructose 1,6-biphosphate and some books will say fructose 1,6-diphosphate. Don't get confused. You know, people just say things differently. Now, the reason for this stage, the importance of this stage is that glucose on its own is pretty reactive. However, the addition of these phosphate groups to it actually makes it more reactive. So it's more readily broken down. It'll be ready for action during this whole process. That's one reason. The second reason, because this is occurring in the cytoplasm, as we know, the glucose molecule is one that can easily pass through into through the cell membrane of the cell and leaves the cell at will by diffusion. But addition of these fructose to these phosphate groups to the glucose actually prevents it from going through the cell membrane. Okay, so those are the 2 main reasons. But I think for respiration purposes, the main reason is that it activates the glucose. Since I call it the activation stage of glucose. All right. Now the next part we are looking at is the splitting of the hexose into the hexose biphosphate. I said hexose, hex means 6. So remember it's a 6-carbon sugar and you have our first breaking of bonds. Now I want you to think about this. Each time a bond is broken in your original glucose, you are looking at the release, the possible release of energy each time. So there is one split down the middle, and when that split occurs, you are making glyceraldehyde 3-phosphate. I'm sure you remember that from photosynthesis. That's the first carbohydrate made by photosynthesis in Calvin's cycle. And so here we have it coming back in respiration. So it is made, just to let you know, you will see in your books as well, that this other substance, dihydroxyacetone, is also made, but it's not used in the process of respiration. It does not continue along in that for the process of glycolysis. So it is easily converted by a next enzyme to glyceraldehyde 3-phosphate. So eventually, both molecules will become glyceraldehyde 3-phosphate. Again, as you can see, the phosphate groups are still attached, one on each molecule, because it's just split in the middle. So just all we have is a splitting of the glucose molecule, so you end up with 2 of them. Please remember to keep the carbon numbers in line. Just you need to remember that. Now the third step. The third step is about the dehydrogenation of the glyceraldehyde 3-phosphate. Dehydrogenation, if you remember, is to remove hydrogen. And also, if you remember clearly from photosynthesis, I also said to you that anytime a substance is, a hydrogen is being removed from a, from a substance, it's being oxidized. And so, and then anytime it accepts hydrogen, it is being reduced. So what happens here when dehydrogenation takes place, what happens is this substance, NAD, now it looks like the one we use in photosynthesis called NADP plus, but no, the P is not there. For respiration, it's NAD, and NAD plus accepts the two hydrogens that's coming off of the phosphoglyceric acid, and that is basically, okay, the phosphoglyceric acid. And so what happens is that the NAD becomes reduced and each of the original phosphoglyceric acids will be reduced and lose their some hydrogens, and that will go to NADH. Now, NADH, just like the NADPH of photosynthesis, has a similar situation in that later on it will become important for the whole process of respiration. Now, during this whole process of the dehydrogenation, what happens here is that ATP is also is produced. So two of them is produced per phosphoglyceric acid or glyceraldehyde 3-phosphate. But remember, there were two of them originally. So at the end of respiration, or glycolysis, that is, you end up with 4 ATPs being made. Now, these 4 ATPs that are being made is not, obviously, you would not call that the total yield of glycolysis, because at the end of glycolysis, pyruvate is made, and I'll explain why this is saying pyruvate and this one here is saying pyruvic acid. I'll explain that shortly. But what I want you to grasp is the fact that the ATPs, even though there are 4 of them in total that is made, you have to remember at the start of the process, we had invested 2 ATP at the start. So therefore, even though 4 is being made at the end, we still call it a net yield of 2 ATP molecules, okay? Because you invested 2 at the start, so even though you make 4, the 2 of them is counted as well. That was your investment in the first place. Now, there's a series of reactions that takes place between the phosphoglycerate that was made before to the pyruvic acid that is made at the end, but we do not have to go through each individual step. What you need to get is that the fact that the ATPs that are being made, the making or the reduction of NAD plus to make reduced NAD, and also the fact that pyruvate is made. And you remember it's a 3-carbon compound, 1, 2, 3 carbons that is here, which tells us that there are 2 of them that will be made per glucose molecule that started the reaction in the first place. Now, just to let you know, in some books you're going to see pyruvate, and in some books you're going to see pyruvic acid. Now, pyruvic acid is really what is made at the end. However, IRL, you find that because there's a water environment that all reaction goes on in, there's always ionization of an acid in water. And so the anion of the pyruvic acid is pyruvate. All right? So there are several intermediate steps that takes place before you reach to the end of glycolysis. And notice, so far, no oxygen is required for this stage. And that is the reason this stage deals with what is called anaerobic respiration, which is respiration that occurs without the use of oxygen. But as you can see, the max total yield of anaerobic respiration is only 2 ATP molecules. Clearly, that would not have been enough to sustain life for larger organisms. Small, like bacteria and so on, they might be able to handle little 2 ATP per glucose molecule, but not big organisms like our cells and trees and so on. Okay, so we'll talk more about it later. Just an overview of what we were speaking about earlier of glycolysis here, the investment of the 2 ATP at the start, the splitting of the glucose or the fructose phosphate molecule, and the making of the glyceraldehyde 3-phosphate, as you know, and then after several reactions, the making of pyruvate. And just to let you know is that during that whole process here, that reduced NAD is made, and there are 2 of them, 1 per molecule of the glyceraldehyde 3-phosphate, and there's also 2 ATP per molecule, which give you a total of 4 molecules. And so far, as you can see, only what has happened to glucose, it has been broken into 2. That's it, just broken into 2. And the yield, as we went through already, because they ask this on exams all the time, so there's no way of getting away from not knowing this, is that you end up with 4 ATPs in total. However, it's a 2 ATP net, very important. 2 NAD is formed, or reduced NAD. I like to use the word reduced NAD as a purist in terms of chemistry. And 2 pyruvate molecules are formed. Please note, they are both 3-carbon compounds, so we have not lost any carbons yet, so there's no CO2 being produced or anything like that just yet. All right? So the number of carbons we started with is 6. Number of carbons we have ended with is still 6. And the number of carbon-carbon bonds that have been broken is 1. And so far, we have only yielded a net of 2 ATP molecules. All right? Now, the next step after glycolysis happens, if there's no oxygen, the process goes into going into what's called anaerobic respiration. Now, the purpose of this process is so that the NAD, the reduced NAD that we spoke about earlier, that was made in glycolysis, can be regenerated or be oxidized to become NAD+, so that it can be fed back into the whole system of glycolysis in the earlier stage. So what happens, depending on the cell, if it's a lactobacillus cell, you have what's called 2 types of anaerobic respiration, main types. There are others, but we don't want to talk much about them. The 2 main types are lactic acid fermentation and alcoholic fermentation. Lactic acid fermentation is what occurs in our muscle cells, skeletal muscles, okay, only in our muscle cells, and that is only for a very short period. And it produces lactic acid. Now, lactic acid is also a 3-carbon compound. So there's no CO2 that is produced during lactic acid fermentation from the pyruvate. In alcohol fermentation, what is made is, it usually occurs in like yeast and other microorganisms. What is made is, the alcohol that's made is called ethanol, which a lot of people are familiar with, and a lot of industries has made a lot of money on alcoholic fermentation. And it also, because ethanol is a 2-carbon compound, carbon dioxide is also produced. And hence the baking industry, because of that fact, makes a lot of money again using that whole premise of alcohol fermentation, because the CO2 is released during that fermentation and causes our buns and bread and so on to rise. Okay, so that is the whole thing about alcohol fermentation and lactic acid fermentation, which all has to do with anaerobic respiration. This is respiration without the use of oxygen. And I'd just like to let you know that during the process, after pyruvate is made, the ethanol that is produced or the lactic acid that is produced does not yield any more ATP. So all of the ATP that has been made from this respiration occurs during glycolysis. All right? So now we need to go to the next stage, because what happens is that if there is oxygen, then aerobic respiration will take place, and this occurs in the mitochondria. All right? And so it's important that we know what the mitochondria looks like. So it is less complicated looking than the chloroplast did, but it's about the similar size, can be about the similar size to the chloroplast. It also has a double membrane, as you can see here, 2 membranes. It also is like the chloroplast, that is. However, the inner membrane is folded in, heavily folded. And that heavy folding is called a cristae. For the mitochondria. The mitochondria also has its own ribosomes and DNA. Anytime you see that in an organelle, ribosomes, DNA, those are protein-making mechanism already. So you know that the mitochondria is making its own protein, usually the enzymes that are involved in the whole process of respiration. Okay? So in the exam, this is what you see, the black and white diagram. So I thought it would be a good idea to say, you know what, instead of showing the nice fancy 3D ones that you might like to see and it's easier to recognize things, let's show you the one that you will see on the exam paper. Because a lot of time when students see these things, they're totally bewildered in the exam, and it's important that you familiarize yourself with what it looks like before you go into the exam. And also to learn to ignore some of you see these black splotches and stuff. These are just what we call artifacts in the biology world when we look at microscopy, because what happens in the preparation of the slides, sometimes things ends up in there, and as the scientists, you need to know when it's just foolishness and when it's actual part of the structure that you're looking at. So you need to be able to identify the cristae, the enfolding membrane of the internal part of the mitochondria. So these lines you see here, cristae. Then you need to know the matrix, which is the dead center, the inside part of the mitochondria. So you need to know where those are. Okay, you need to be able to recognize the intermembrane space and the inner membrane versus the outer membrane. Also, we need to know where things happen. The matrix, which is the inside part or the middle part of the mitochondria, that's where we have the link reaction and Krebs cycle, which we'll discuss in a future class. And also in the cristae, the inner foldings, the membranes that are folded inwards, this is where what's called the electron transport chain occurs. All right, class? So we are going to just now look at some questions to help us see what we have remembered so far, and so that we can move on in the near future. All right, I wanted to say something to you about, in the drawings of the mitochondria, please know that in the fancy 3D diagram, you would have been able to recognize DNA and ribosomes and all of that. In this, you're not going to be able to see those things because there's so much inclusions in this black and white diagram that sometimes children mistakenly label things that are not really there. They're just, artifacts or things that you happen to see. So be careful about that when you're doing an exam. All right? So, as I said, we're going to go through our first question. First question is, why is the phosphorylation of the glucose at the start of glycolysis necessary? You should remember that we spoke about it. It prevents the glucose from diffusing out of the cell, and it also energizes the sugar, so make it more unstable and therefore more reactive. Okay? Next, explain the importance of glycolysis taking place in the cytoplasm of all cells. Well, think about it. If all cells All cells have a cytoplasm. All cells don't have a mitochondria. So the mere fact it occurs in the cytoplasm, it means that all cells can respire, both prokaryotic cells and eukaryotic cells as well. Okay? The last question we have is, what is the fate of the products of glycolysis? Now, we didn't discuss that directly, so we can discuss it now. The ATP, as you know, will be used for energy-rich activities and reactions that occurs in the cell and in entire organism as a whole. The NADH, or the reduced NAD, which I like to call it, is going to be taken now to the mitochondria, to the inner part of the mitochondria, to the electron transport chain to release even more ATP. So you see that NAD that you see that has been made, it has use for future ATP. But in the anaerobic section with alcohol and lactic acid fermentation, that was not utilized. The whole fact of using the NAD or the reduced NAD to make more ATP, that was not utilized. Okay? So that's why the aerobic respiration is so important. Pyruvate. The pyruvate that is made, if there is no oxygen, then lactate or alcohol fermentation will take place. However, if there is oxygen, it will then be broken down even further. In future classes, we'll talk about that, where we'll look at Krebs cycle. We'll first look at the link reaction, then we will look at Krebs cycle, and then we will look at the other stage of aerobic respiration, which is the electron transport chain to release more ATP because of the production of reduced NAD. I hope that the whole process of respiration is something that you are going to learn well, because it is a part of the exams, the syllabus, that comes every year on the exam. That module 1 on photosynthesis and respiration, I can assure you, will come. Either a structured question or an essay, long essay worth 13 points or so on. And please learn to relate the whole topic of respiration with photosynthesis. That's the next thing, the key thing. You need to be able to relate the two things together, because what happens is, if you notice at the start of respiration, glucose starts respiration, but at the end of photosynthesis, glucose is what ends it. So you can see one is the reverse of the other. So it's extremely important, class, that we do this, all right?